Sickling test is a diagnostic test for sickle cell anemia. Sickle cell anemia is an inherited disorder in which the point mutation occurs on the beta chain at 6th position where the glutamate is replaced by the valine. We all know about this. And also the RBCs present in this particular disorder, sickle cell anemia, are sickle shaped, right? So because of its sickle shape, it has very less oxygen carrying capacity, and also it can accumulate in different sites leading to the various problems. So before we go on to the next point, let us talk about sickling. What exactly is a sickling? So conversion of biconcave RBC into the sickle shape is called sickling. So this occurs in case of the sickle cell anemia. Under what circumstances this occurs is in case of hypoxic conditions or in case of dehydration or acidosis. Under such circumstances, this normal biconcave RBCs are converted into sickle shaped or the holly leaf type of shape. So the question is, each and every RBC under this deoxygenated conditions get converted to sickle shape? No. Only the RBCs having hemoglobin S are converted to sickle shaped under deoxygenation, dehydration and acidosis conditions. So there is another factor which can induce attainment of this sickle shape is by exposing this blood to sodium metabisulfate. So sodium metabisulfate is a reducing agent which induces the attainment of sickle shaped in cells of in RBCs of sickle cell anemia which have HbH type of hemoglobin. It gives an holly leaf-like appearance. Remember this point. So principle, sodium metabisulfate reduces the oxygen tension in the RBC. That means it creates an hypoxic conditions in the RBCs, and the RBCs which have HbS type of hemoglobin under this conditions get converted into sickle shape. So this is how we can know if a person is having sickle cell anemia or not. So the procedure to do this test is add 1 drop of blood and 2 drops of sodium metabisulfate and observe it under the microscope. This is how you find under the microscope. These are the sickle shaped cells. So this is very narrow, and as you can see there is less oxygen carrying capacity, and this sickle shaped cells can accumulate in the various sites, can lead to hemorrhage, can lead to splenomegaly, it creates many problems. So this is the holly leaf. Sickle cell RBCs resemble this holly leaf, right? So that's why we call it as holly leaf appearance under the microscope. And other diagnostic tests for sickle cell anemia are first one, sickling test we studied here, and solubility test for HbS, hemoglobin electrophoresis. So this different types of hemoglobins are made to run on agar. HbH is lower than hemoglobin A and fetal hemoglobin. And the fourth diagnostic test is estimation of fetal hemoglobin. So why is it done? Is because fetal hemoglobin inhibits sickle cell anemia. Fetal hemoglobin is decreased in case of sickle cell anemia, right? This is a diagnostic test for sickle cell anemia. This is about sickling.